volatile or can be made so. One important use is to determine whether a newly prepared compound is contaminated by solvents. The separation takes place inside an oven which is used to maintain a controlled temperature. Samples are usually stored in a carousel where they stay until they're placed in the sample changer. Each sample is injected into the gas chromatograph here by a syringe. The handling of the samples and storing of data is automatic and controlled by a computer. Inside the oven is the capillary where separation takes place. A typical capillary tube may be 25 meters long with an internal diameter of 250 micrometers and is often made of silica. The inside is coated with a liquid stationary phase. An inert gas such as helium carries the sample mixture through the capillary. Parts of the sample will show a greater affinity for the stationary phase and travel more slowly. Over the 25 meters, the components of the mixture become separated along the capillary tube. At the end of the capillary, the separated components are carried into and through a detector. This instrument has a flame ionization detector. Each component in turn is fed into a hydrogen-air flame where it burns, producing ions. The ions allow a current to flow between the two electrodes that surround the flame. Gas chromatography uses about 1 milligram of sample dissolved in a volatile solvent at a concentration of about 1 milligram per cubic centimeter. Once the details of the samples are entered into the computer, an automatic sample changer carries each sample in turn to the instrument. A small portion of the sample is drawn up into a syringe and then rejected to rinse the needle. About 1/1000th of a cubic centimeter of sample solution is injected into the instrument. The needle is then rinsed to leave it ready for the next sample. Each peak on the chromatogram represents a component in the mixture that has been separated in the capillary tube and detected in the flame.
High-performance liquid chromatography, usually called HPLC, is a powerful technique capable of separating very similar compounds, even pairs of optical isomers. In this instrument, up to 4 separate solvents can be used for separation. The mixture is pumped under high pressure through the separation column. Absorption of ultraviolet radiation is used to detect the separated compounds at the end of the column. The process and the storage of data are controlled by a computer. Inside the instrument, the separation takes place in a column packed with tiny particles of porous silica. A mixture of solvents, the mobile phase, is forced by a high-pressure pump through the tube of silica, the stationary phase. Different compounds move at different speeds through the tube, depending on their relative affinities for the two phases. The compounds become separated. Different columns with different stationary phases are used for separating different types of compound. The reservoirs of solvent sit on top of the instrument. This instrument uses 4 solvents that can be mixed in any proportion. The composition of the mixture can be gradually changed during the run. Dissolved gases are removed from the solvents and pumps force the solvents through the column. The pressure difference across the column is about 13,000 kilopascals. This is the flow of liquid directly from the pump. The rate of flow through the column is set at 1 cubic centimetre per minute. From the column, the solvent and the separated samples pass through a detector. This shines ultraviolet light of a range of wavelengths through the samples and detects any absorption. The sample is made up at a concentration of about 1 milligram of the solid material dissolved in 1 cubic centimetre of solvent. The bottle of sample is carried to the instrument using an automatic sample changer. About 1/100th of a cubic centimetre of the liquid sample is drawn into the instrument by a hypodermic syringe. With some instruments, the sample can be injected by hand. A computer controls the operation of the instrument. The computer will also store the data from the run and produce this in the form of a chromatogram.
Gel electrophoresis is a technique that is used for separating mixtures such as proteins and RNA found in cells and estimating their amounts. It can measure quantities as low as 1 picogram, 10 to the negative 12 of a gram. The proteins are treated to make them negatively charged. In an electric field, the larger protein molecules move more slowly through the gel and become separated from the faster-moving, smaller molecules. In this apparatus, there are 2 separate layers of polyacrylamide gel, each 3/4 of a millimetre thick. These 2 cells are identical. Each sandwiches the gel between a glass plate in front and an alumina sheet behind. In this case, only the front cell is to be filled. The layer of gel between the plates is topped up with liquid gel. A plastic comb is used to leave wells in the gel as it sets. A buffer solution makes the electrical contacts between the gel and the electrodes. The colourless proteins are treated with a mixture containing glycerol, bromophenol blue, and sodium dodecyl sulfate. The bromophenol blue colours the mixture. The protein molecules absorb the dodecyl sulfate anions and become negatively charged. The plastic sheet marks the position of the wells in the colourless gel. The protein mixture with the added glycerol sinks in the wells left in the solid gel. This darker blue solution is a rainbow mix made specially to show the separation. It is a mixture of different proteins, each coupled to a differently coloured dye. The electrophoresis unit is covered and connected to a power pack. Circulating water keeps the unit cool. For this separation, the current is set at a constant value of 16 milliamps. The thin blue band is the bromophenol blue dye, moving ahead of the negatively charged protein molecules. Normally, this is all that can be seen. The darker blue is the rainbow mixture, which separates into its different colours. They're normally rather pale, and the colour of these lines has been enhanced to make them easier to see. The colourless proteins have to be transferred to a sheet of nitrocellulose to prevent them diffusing through the gel. The gel is slid from its backing sheet onto blotting paper. The transfer is completed by sandwiching the layer of gel and the nitrocellulose together in a frame. They are held in contact between sheets of blotting paper and layers of foam. The package is placed in a transfer tank which is water-cooled. The tank itself is filled with electrolyte. An electric field is applied across the layers. The large protein molecules, coloured yellow in the diagram, move to the surface of the nitrocellulose. The small blue dye molecules pass through the nitrocellulose and the blotting paper to be absorbed by the foam. The nitrocellulose with the protein molecules is treated with reagents which colour the proteins. The dark lines mark the position of the proteins that have been coloured. The bands of colour are from the rainbow mix, which shows that the separation has taken place. The amount of protein is estimated by measuring the density of the coloured line on a densitometer. A spot of light shines onto the nitrocellulose. The dark-coloured line absorbs some of the light so that less passes to the detector. The quantity of protein is related to the amount of light absorbed and to the area under the peak of the curve.